welcome to the online lecture again. Today we'll talk about DNA structure, replication, and expression. DNA is a very important topic because it is universal in all living organisms. Okay, structure of DNA. It was first discovered by James Watson, Francis Crick, and Maurice Wilkins. And they got a Nobel Prize for discovering the structure of DNA. Maurice Wilkins is like a lab tech. James Watson at that time, he was like a grad student for Francis Crick or something like that. There's some side story to it, but we first cover this. DNA is a chain of nucleotides. Remember at the beginning of this semester, we talked about the four major macromolecules, and DNA, nucleic acid, is one of those. And the monomer for DNA is called nucleotide. And DNA is a polymer. So the DNA structure, the nucleotide is composed of these three subparts: phosphoric acid, the phosphate group, a pentose sugar, the deoxyribose, which is a sugar, then nitrogen-containing base, alternatively nitrogenous base. These we'll talk more about in details in the next few slides, but I want to give credit to someone before we move on. It is, her name is Rosalind Franklin. Rosalind Franklin, she is a female scientist. Many people believe that she should deserve to get the Nobel Prize instead of Maurice Wilkins, because Maurice Wilkins is just a lab tech. She is actually the first scientist who is able to take the X-ray structure of DNA. At that time, this technique is called X-ray crystallography. And what happened is that she's able to take the X-ray of DNA, but in order to decipher or to crack or to solve the DNA structure based on the X-ray crystallography, it takes you need to be really, really smart in order to do that. It's not everyone can do it. She is a very good scientist who can take the X-ray crystallography, but she is not able to. After she took the X-ray, she just put it in her lab. And then, according to the story, Francis Crick and James Watson asked Maurice Wilkins, the lab tech, to so-called borrow her X-ray crystallography without letting her know. So Maurice Wilkins went to her lab and get the X-ray crystallography film, and then James Watson and Francis Crick they look at the film, and then after they look at the film, they start to try to figure out the structure of the DNA. And according to the story, James Watson he has a dream, and then in his dream he figure out the structure of DNA is the double helix. And then after that, they got the Nobel Prize. Unfortunately, without her. So I want to give credit to Rosalind Franklin here. She should deserve the Nobel Prize. And this is James Watson and Francis Crick. This is James Watson. This is Francis Crick. And this is James Watson. I believe it's 2011. And this is me. Yes, I met him in the Cold Spring Harbor Laboratory in New York, Long Island. He was the chancellor of the Cold Spring Harbor Lab. So, structure of DNA. There are four possible bases in DNA. Two of them are we call purine A and G. Adenine and guanine, two pyrimidine, thymine, and cytosine. So I usually arrange them in a table like that, like this. So I like to arrange it like this, right? So remember at the beginning of the semester, I gave you a way to memorize at Gentleman's Club. But as I said that some people like to memorize it at a Gentleman's Club, Gentleman's Club. Because A pair with T and G pair with C. So remember that. And then DNA, as I told you, as we know at the beginning of the semester, is a polymer. And here we have polynucleotide because DNA is made of many nucleotides joined together. So you can discuss with 2 or 3 students. Well, since it is our 9th lecture, you should think about this. Which molecule you have learned from this class has or have the most similar structure as nucleotide? So you can think about this, and I'll ask you the same question on Wednesday's lecture. Two strands make up double helix. Yes, hope everyone knows that the DNA structure is a double helix. And the two strands are held together by a hydrogen bond. So remember, the two strands of DNA they are held together by a hydrogen bond. And as I told you that A pair with T and G pair with C. And the two strands we call them anti-parallel. Anti-parallel because they are in the opposite direction. What do we mean by opposite direction? We indicate the direction of DNA in something called 5' and 3'. So you can see here, there's a 5' and a 3' of the DNA strand. So what do I mean by that? So if you look at this is a nucleotide. The nucleotide is a subunit of DNA, it's a monomer of DNA. Here's a phosphate group. Here's a pentose sugar. That means a sugar with a hexagon shape, and it has a nitrogenous base here. And as you can see in this picture here, we number the carbon atom on the pentose with 1 here. So the carbon in here is number 1, 2, 3, 4, and then we have a carbon here, we got 5. And this point here, it is not a carbon, it is an oxygen atom. And on the opposite strand, you can see that it is reversed. You can see that here. Meaning that the 3' end is on top, the 5' is coming downward. But here, the 5' carbon is pointing upward, the 3' carbon is pointing downward. Because of that, we call it one strand is 5' to 3', the other strand is 3' to 5'. And as you can see that A always pair with T, and G always pair with C. And between A and T is always, it has 2 hydrogen bonds. And here, G and C it has 3 hydrogen bonds. So we can add in here 2 hydrogen bonds and then 3 this is the double helix structure. So after the polyethylene, you twist the entire molecule, and it becomes a double helix structure here. Because double helix, because one strand will form a helix, the other strand will also form a helix. So they wind together. So this is another question that I'll ask you on Wednesday. Which molecules have you learned from this class have the most similar structure as DNA? Now we finished the DNA structure. Let's go to the second part of today's lecture, DNA replication. DNA replication is the process of copying one DNA double helix into two identical double helixes. In other words, DNA replication is like making a photocopy of a DNA. But it is, and where have we learned about DNA replication? We learn about DNA replication in mitosis. Remember the cell cycle that I told you? In the S phase, S stands for synthesis. In the S phase, what happened? DNA replication occurred during S phase. So this is what we're going to talk about, the DNA replication in the S phase. And DNA polymerase is the key enzyme for the DNA replication. So you need to know the name of the enzyme. So ASE means it is an enzyme. Polymer, well, you know what a polymer is? DNA. So what is the function of this enzyme? It is an enzyme that makes DNA polymer. So it basically it connects the nucleotide together and then makes the DNA polymer. So this is the function of this DNA polymerase. And then there's another type called semi-conservative. Semi-conservative meaning that it uses the original strand as a template to produce a complementary strand, or the daughter strand, you can call it a new strand. So I'll show you a picture later what a semi-conservative means. So I can draw it out what semi-conservative means. So this is DNA strand. Now we have the base pair here. And then the new complementary strand will be here. We call it semi-conservative because for this new DNA molecule here, this part, we have the new DNA molecule here. This new DNA molecule, we have one strand that is old. The other strand, it is new. So we call it semi-conservative because one strand is old, one strand is new. And the resultant, we have two new daughter DNA molecules. We have two new daughter molecules, and they will be exactly identical to the original one, the parent molecule. So it says that before before replication occurs, the two strands of DNA, they are hydrogen bonded together. So we have to we have H bond here. And then this is another enzyme that you need to know the name, DNA helicase. Unwinds or unzip the double-strand DNA, that means in here. You see that the DNA, they are together, and then originally is a double helix, and then this helicase will bind at the beginning of the split here to unwind the double helix. So the name of it is like ASE, meaning that it is an enzyme. Helicase, helix. So you know that DNA is a double helix, that means it is twisted. So in other words, this enzyme unwind the DNA helix. And this is the function of it. So what it says in the second point here is that we have DNA helicase to unwind the DNA here, and then we have a DNA polymerase which will move along the parental strand. The parent strand will move the blue strand. Then as it moves along, then you will grab the free nucleotide, the free nuclei, and then put them to match the parental strand here. And so this is the way to replicate DNA. And then after the polymerase move along the mother strand and put the nucleotide, the free nucleotide, to match the mother strand nitrogenous bases, then we have the third enzyme that you need to know is called DNA ligase. What it does is that after DNA polymerase put the free nucleotide to match the mother strand, you have they are not connected at that time. So what happens is that its DNA ligase will connect all these free nucleotide so that it will become a continuous strand. And after that, the replication process will come to an end. So this picture basically summarizes what's going to happen. So you see that the parental strand, and then these are the daughter strand. You can see that it has one old strand from the parent and then one new strand from the daughter, same as here. So we have two daughter strands, they are identical. That means the sequences of the nitrogenous base will be identical to the parent strand. But we call it semi-conservative because one strand is old, one strand is new. And you can see that, okay, this is a double helix. The opening of the unwinding and opening of the double helix is done by the helicase, DNA helicase. And then each strand will be bound by a DNA polymerase. The DNA polymerase will keep adding this free nucleotide according to the base pair to make the new daughter pair. But as you can see that when it first added, they are not connected. They are not connected. So what happened is that you need the third enzyme, DNA ligase. The connect the three nucleotides so that they will become a continuous strand. And as you know, there's something that you need to memorize, which is very important, is that, the direction of replication. So for the green daughter strand, the DNA replication, the daughter strand always replicate from 5' end to 3' end. This is something that you need to memorize. So, as you remember, the parent strand, they are anti-parallel, meaning that if it is 5' end here, this will be 3' end, and 2 will be 3' end here, this will be 5' end. And the daughter strand, the new daughter strand will also be anti-parallel. That means this will be 5' end, this will be 3' end, and this will be 3' end, this will be 5' end. And according to this rule, the replication direction will be from 5' end to 3' end. So what happens is that this one, you will replicate from this direction. The replication will be from this direction. You can take a look at here. You can see that one strand is going from this way, but the other strand is going into the fork. The one that goes into the fork, we call it the leading strand. This one is the leading strand. This one we call it lacking strand. So what is a leading strand? What is a lacking strand? Remember, the leading strand is continuous. But the lacking strand, they are properly in the fragment. The fragment, we call that Okazaki fragment. So because of the Japanese scientist, Dr. Okazaki discovered that, so he named it after himself. And then, because you have this fragment here, you also need DNA ligase to connect all these Okazaki fragments in order to get a continuous strand of DNA. So let's watch a video.
Like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides, which are hydrogen bonded to each other to form a double helix. nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the antiparallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase, because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, 2 identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
Okay, in this video, it tells you more than just 3 enzymes. But for our class, I only require you to know 3 enzymes, namely DNA polymerase, DNA helicase, and also DNA ligase. So I only require you to know these 3 enzymes. With gene expression, let's talk about gene expression in the next video.